Hello, my name is Tori Hale, and today I would like to focus on cell cycle regulation. Now, at this point, I'm sure you're very familiar with the cell. The complexity of it, all the organelles that are involved in just one cell, and obviously we as human beings are made out of a ton and a ton of cells. But when we think about it, how are these cells regulated? What cycle, so to speak, is going on to ensure that when I grow or when I need cells, that I will have them? Not only have them, but have them fully functioning. So before I even dive into the things that regulate the cell cycle, it's first and foremost important just to understand specifically like how many cells were we to begin with? Because if you think about it, initially it was a sperm and an egg, and you formed one cell. And then out of that one cell, you get the complex human being that you and I are today. In fact, Caldwell put it this way: the process by which one original cell carves out such brilliant and varied feature sutures, its progeny has been an enigma, easy to observe, impossible to explain. I love how he said easy to observe and impossible to explain. Because when you think about it, especially in the 1990s, we really did not know, especially in comparison to today, what we know about the cell. Because we knew that they were complex, that they were differentiated, that it allowed us to be the human that we are today. But to explain that process seemed to be a blur. And even today, we're learning more and more about how the cell cycle truly functions, how our cells function, and how ultimately we are the human that we are today. Now, two key breakthroughs came out in regards to explaining this so-called impossible explanation of us in cells. And that specifically was DNA structure and something called the homeobox. Now, if you look at the picture, you'll see that there's a wild type of this Drosophila slide, and you'll see that the antennas, right? They look normal because they are on the wild type. However, if you look over at the mutant, you'll realize that what ultimately should be antennas is actually where legs were placed. So literally the fly legs replace the antenna. Why is that so? Well, that's because of this idea of homeobox. All right, homeobox got its origin because these were first studied in these homeotic genes that were gathered together in clusters. And they realized that according to the DNA sequence, they were clustered together to mimic the order of the body parts that should be in a quote-unquote finished fly. And so ultimately, this homeobox kind of helped depict how we are born and differentiated, why we have cells that create our antenna, why we have cells that create our eye, legs, so on and so forth. And specifically, if I could circle back to this idea of DNA structure, when we finally got a glimpse of what the DNA was all about and enabled us to figure out why, someone has red hair or brown hair, or, why we have this genes that we do. And just think of it today, we have the opportunity to send in a swab, come back and figure out what types of genes that we have, what we're, more predisposed to, are we, going to have Alzheimer's in our futures? All of that is detected through our DNA, which again is comprised in our cell. So you got to think, if I'm made of this one cell that obviously made me, how did that happen? Because clearly there has to be some type of cell cycle regulation. Why do I say that? That's because if we look at the cell cycle and we go to its basic structure, it tends to occur in four main stages. Number one, the G phase. So we got the G phase, we got the S phase, we got the G2 phase, and we got the N phase. Now, why are these stages specific? Well, first off, if we look at the G1 phase, this is where your cell is going to increase in size. It's where your cellular contents are going to be duplicated. So again, G phase, I tend to allow my students to think of it as a growth phase. So this growth 1 phase is when my cell is going to increase. But the whole reason is why. Why is it going to increase? because ultimately your cell needs to divide. And how can I divide my DNA and ultimately replicate it if I don't have enough nutrients in it? So it has to grow first before it enters this S phase. So we enter the S phase, that's where your DNA starts to replicate. Again, you have about 23 pairs, 46 chromosomes that are ultimately replicated by that cell. Then we enter into the G2 phase, which is after synthesis. That's where your cell again is growing because it's preparing for the ultimate division, which is going to occur in this M phase known as mitosis. So as you can see off to my right in this circled region, again, we have this growth phase. Once it grows enough, it can begin to synthesize. Then it continues to grow because it's got to be ready for this mitotic division. And obviously, since we were one cell and now are made of many, all right, this mitotic process constantly occurs. Now, obviously, when you're in the womb and you're growing all these new bones, all these new cells and everything like that, of course, you're gonna have a ton of mitotic division. But even as adults, little people think, oh, why do I need new cells? Well, we constantly, even in our skin, shave cells off of us, so we constantly need to replenish it. Even if we think of bone cells like osteocytes, if there's any type of friction that occurs between the joints, guess what? I'm gonna need new cells. What if my bone breaks? Again, I'm gonna need cells. So we're constantly, even as adults, generating and having this process of mitosis occurring in our cells. So this is just a basic overview of the cell cycle and the four main stages of it. Now, let's look a little bit in more detail so we get a better glimpse of mitosis. So again, we have G1, S, G2, and this leads us into the mitosis stage. Again, thinking of the complexity of it, mitosis is just not one step. In fact, there's 4 general steps that are associated with it. You have the classic prophase, metaphase, anaphase, and telophase. Now, if you're like me, you definitely learn better by not only talking about it, but looking at pictures. So if I think about it, we can zoom in to prophase right here. This is when your nucleolus is going to disintegrate, your nuclear membrane is going to break down, your spindle fibers appear. And then from this idea of prophase, all right, you're going to get what we call prometaphase, which is that in between. Now specifically, as you can see, we start having my chromosomes attached specifically to the spindle, and ultimately that's going to lead to 4, which is way over here, metaphase. So metaphase is the actual process where the chromosomes start to align in the center so that they're ready in the anaphase portion to completely divide. So again, just to reiterate, prometaphase is when the spindle fibers attach to the chromosomes condense. Metaphase, as we see, it begins to align. A lot of times I tell my students, M for middle, if that will help them to remember it. Then we have anaphase, where they break apart. So I got my A's there. And that's also where your sister chromatids begin to move to their respective sides. That leads to telophase, where your nuclear membrane reforms, the chromosome decondenses, your spindle fibers disappear, and then ultimately will lead to cytokinesis, which again, if you remember your prefix cyto means cell, kinesis movement. So that's really where that parent cell ultimately becomes 2 daughter cells. So again, why in the world am I even taking time to mention mitosis? It's because if I scroll all the way back up to the previous slide, if I slide all the way back up, what are we talking about? The cell cycle. And how can I understand the things that regulate the cell cycle if I'm not understanding all the processes that are occurring? So again, this is much more complex than a lot of people like to say. Now, again, you might say, so what? Your cell divides. Seems pretty simple. Well, I wish that were the case, because guess what? According to research, and I'll state it directly from Cooper, he stated that the frequency of errors during replication correspond to only 1 incorrect base per 10 to the 9th to 10 to the 10th nucleotides incorporated. Now again, that is extreme accuracy. There's only one incorrect base out of 10 to the 9th to 10 to the 10th nucleotides. But you also got to think of it this way. Not only has God given us an intense proofreading system to again help remove those small incorrect bases, but we're constantly again undergoing this phase of mitosis. So you might just think, oh, one incorrect base for 10 to the 9th. Still, could those mutations be harmful? Yeah, and lead to even cell death like apoptosis, or just an innumerous amount of it and lead to cancer? Absolutely. So it's important, very, very important, that we understand that we need cell cycle regulation, because even in the replication process, errors can occur. And again, errors in this case can lead to something as catastrophic as cancer, or in this one example, this idea of neoplasia with the vulval intraepithelial neoplasia, or otherwise known as VIN. So even just looking at this, I see my normal cell developing, but guess what? When there is an error in the cell cycle, it can lead to an innumerable amount of abnormal cancer cells. And sure, some of it may be benign, just the simple development of it. However, many of those could develop into an invasive cancer, potentially even metastasize and be a very, very harmful to a human, obviously even leading into death if not taken care of appropriately. So it's important that our bodies have this type of cell regulation, because between all these processes, the G1, the S phase, the G2, the M phase, what if your cell multiplies and it forgets your DNA? What if your cell multiplies and it didn't have enough nutrients? What if your DNA attempts to multiply, but you don't even have room to grow because you have not even taken the time to really grow in those G1 phase and that G2 phase? And that's why God has innately developed us and given us these checkpoints, so to speak. And this is really what I want to harp on in regards to cell regulation. It's these key checkpoints in the cell. Now, there's 3 specifically that I want to touch on today. But again, it's much more complex, as you'll see through some of my pictures. But ultimately, we have the G1 phase, otherwise known as the restriction point. If you look at it on the picture to my right, you'll see it right here that I just highlighted in yellow. So at the end of G1, I enter this G1 checkpoint. Makes sense. I'm checking G1, thus G1 checkpoint. Additionally, I have my synthesis page. So of course, I need a G1 checkpoint to make sure it's grown enough so that I can begin synthesizing my DNA. So I go there, I get the cell growth. Okay, that's when I get into my G2 checkpoint, which I'm going to touch a little bit more about. Again, at the end of that. So at the end of G2, right before I head into metaphase, I'm having this checkpoint to make sure, can I go on to the next step? And then last, this idea of the M checkpoint, the mitotic checkpoint. So let's talk a little more specifically about each individual checkpoint. But before I do, I want you to think of an example. So because I'm a teacher, for example, if I could be teaching like physics, and I could be talking about creating a catapult. It would be very catastrophic for me as a teacher to say, okay, create the catapult, just read it and then turn it in, by 3 weeks. Why? Because most likely people are going to procrastinate and not follow directions and end up with a very, very, very terrible result. That's why, if you think about it and you're a good teacher, you're going to provide a rubric, a series of checkpoints to assure that, hey, my student is following the right path. Did I use the right number of materials? Did I create the proper calculations? Did my projectile go the proper trajectory? Did I do my calculations right? Did I put it in proper APA format? So I have all these small checkpoints to tell the student you're on the right track, or we need to go back and make sure that this is fully completed. It's the same way in our cell. You're gonna, before you wanna synthesize and develop, you're like, am I ready for that yet? all right, if I'm not, I need to stay here. But if I am ready, I can go to the next step. Because if your cell were to wait to check itself until after the division, that's where it's catastrophic. That's where you have that innumerous cell division, those oncogenes, those tumor suppressors that will ultimately lead to some cancerous cells and potentially detrimental to the human that's impacted by it. So on that note, let's turn to the G1 restriction checkpoint. Now, I want to give big objectives for each of these checkpoints so that you understand the big idea. And again, it's all logical. You want to think logical. It's not just something that I'm going to memorize and hope for the best, right? There's reasons behind every checkpoint in the cell. Because again, it's so evident, even from what I've already discovered, that we were created by an intelligent designer. How can this all occur? Again, it's very, very logical. So, when we get to G1, again, we're just talking about the basic growth of the cell before DNA synthesizes. So before DNA synthesizes, the big objective for G1 is to make organelles and proteins. All right? So when I think about this, I want to make organelles and proteins. That makes sense if I'm going to divide, right? Or if I'm going to synthesize my DNA, I need those organelles. I need those proteins to help me do it. Barnum and O'Connell in their study talked specifically about the G1 in budding yeast. So again, there's been a lot of research on this, specifically probably the most popular research on the budding yeast about the control of the G1 cell size checkpoint, specifically with cyclin 3, which activates it. So in regards to this restriction checkpoint, it's been heavily studied, especially in yeast. But specifically, if you think about it, or at least I would be thinking in my head, how in the world does your cell decide to go from one point to the other? So like, for example, when we go back to my teacher analogy, how do I know if my students are going to go to the next step? Well, they're going to look at step 1 and they're going to think, did I complete this, right? And I'm telling them, hey, you met the mark, continue on, or hey, you did not. So who is that teacher in the cell telling it, hey, you need to continue on to your next checkpoint, or you need to hold on because we're not quite ready for the next step? And the thing that really does that is this cyclin expression, the CDK and the cyclin. If you look at the picture, I specifically mentioned and through this chart show which cyclins heavily expressed, which ones not heavily expressed throughout each cycle. So you can see as you go through different checkpoints, G1, G2, and the M, you'll notice that there's a different expression of cyclin and a different expression of CDK. So CDK specifically stands for cyclin-dependent kinases. So guess what they're dependent upon? Cyclin. Now, if you remember, kinases literally phosphorylate things. So CDK may remain dormant, but when reacting with that cyclin, it will actually initiate, right? It will actually cause something to be phosphorylated. So even when I think of the G1 phase, all right, I'm going to see, what am I going to see an increase of? Well, I see this increase of G1 cyclin, and in fact, I can scroll back to our discussion on the other slide. If we look specifically at G1, we see the cyclin D and CDK4. So when cyclin D is associated with CDK4, all right, my G phase is starting to thrive. In fact, then it goes to cyclin E and CDK2. After those, again, are expressed and activated and cause phosphorylation, that's when my G1's gonna know, hey, I can move on. Because there's certain cyclin that, again, maybe freeze it in its place, or there's some cyclin that activate the synthesis so that we can grow or that we can divide. Specifically, as we move on to the G2 phase, its objective is going to prepare for mitosis and make microtubules in the cell. Again, Barnum and O'Connell state that this has most heavily been studied in fission yeast instead of the budding yeast from G1, where the CDC25 and the WE1 respond to cell size and nutritional status. So again, if I'm thinking of the G2 checkpoint, why in the world do I care about nutritional status? because I need the cell so healthy with so much nutrients that I can literally divide it in half. Again, if you don't have enough nutrients and you go to mitosis, those cells are dead. They have no nutrients to support them. So again, each cell cycle has purpose. Again, you can even see up there about how cyclin B and CDK1 are initiated. And this enables G2 to move on to and allows the cell to go from the G2 phase to mitosis. Now, again, I am talking in very general terms at this point, so I want, again, to get across this big idea of regulation, but I also want to point out that even as you see from this, it is highly complicated. You have various things working on. P53 is a very popular one. You'll see a lot of CDCs. You see CHEK2, CHEK1. Those are very common when you read in cancer articles of what's associated with cell cycle regulation. So this is by no means simplistic, but I want to get, again, just a foundation to realize what's all involved with cell cycle regulation. So going on to that leads us to the M checkpoint, the third checkpoint that I want to talk about. And this big objective is just to make sure the cells are ready to divide. Because again, you're in the middle of mitosis. You want to make sure that this cytokinesis can occur properly and that the cells completely separate. Now, in a cancerous cell, they're going to thrive in this mitotic state. They're going to love mitosis because cancer in and of itself is the development of abnormal cells. Whereas the majority of cells tend to rest in interphase. Now, in regards to this, after the M checkpoint, once it passes this, once it thrives and gets through the M checkpoint, then obviously the cell's okay. Again, it's like the teacher gave them the go-ahead, and you have two fully functioning cells. But again, how did I get to those two functioning cells? It was a very complicated process. One cool thing that I always like to share is so what? So I understand the cell cycle. Why does it even matter? One really cool fact about cell cycle regulation is that by understanding it, we can identify a lot of cancers. Specifically, I spent some time studying the CHEK2 1100 deletion of cytosine, which, again, if you remember, I barely stated it in an earlier point. CHEK2 is actually one of the control genes that help with cell cycle regulation. Specifically, it is a tumor suppressor that encodes for protein CHEK2. But when there's a deletion of cytosine on the 1100th nucleotide, it's going to affect DNA repair, cell cycle arrest, and apoptosis. Remembering that apoptosis is programmed cell death. So, by me just understanding the basic knowledge of cell cycle regulation, we were able to dive into this gene mutation and see how it correlates with various cancers. Our studies still need to be supported, but much research, even just our limited amount of research with this particular gene mutation, showed a very high association with potentially prostate cancer. And you can check out that published paper in the references at the end of my slide if you're interested in learning more about it. But it's cool to see that ultimately, cell cycle, just understanding this, it can literally, you could be the next cancer researcher. You can find ways to apply this in so many ways instead of just regurgitating information. And why? It's because our cell's extremely organized, created by an intelligent designer, and it's very easy to predict what's going to happen next. So in summary, let's say you kind of forgot everything that happened and you want a quick snippet of the big ideas. Well, first, you need to realize, obviously, that our body is constantly growing and in need of cells. All right, obviously when you were little, sperm and egg, you had to divide and multiply exponentially. But still, even as adults, we're constantly undergoing this mitotic process to develop new cells. Next, make sure that mistakes can't occur. Why would we have checkpoints integrated in our cells if they didn't happen? So checkpoints are essential. Just like they're essential as a teacher assigning some type of major project, it is so much more essential in the human body. Why? Because the checkpoints are going to limit mistakes. By having this G1, G2, M checkpoint, it limits the possibility of possibly having neoplasia, right? Or in my case, that CHEK2 gene mutation leading to prostate cancer. Because ultimately, if errors occur in these checkpoints, it can lead to cancer. There can be an exponential mitotic expression of cells that are abnormal. Why? Because they didn't follow the basic checkpoints. They didn't listen to the cyclin B, the cyclin D, the cyclin E, the cyclin A, and their association with their specific kinases. So if you want to learn more, feel free to reference these two slides of where I got my information, or specifically my paper right here below. I encourage you to read. But ultimately, it's incredible to see how intricate we are made, the complexity of the cell, and how ultimately cell regulation is a vital part of our life that happens each and every day so that we can live a healthy and normal life.